Welcome, everybody. It is great to have you with us for another webinar from the AAPS Topical and Transdermal community. It's really a pleasure to be able to have you join us today, and we're looking forward to a fantastic webinar presentation, as well as some great questions and dialogue. And so one of the things we're looking forward to is hearing from you and being able to have a chat with you about your questions. And at the end of the webinar, we'll have a Q&A session. In order for you to be able to click in the bottom left corner to queue up, essentially to kind of queue up at the mic, so to speak, to ask your question, you may need to enter the 4-digit code and the hash sign that associates your phone number with your name, because if you don't associate your call-in phone number with your web login, you may not be able to click on that button at the bottom of the screen, and we'd really love to hear from you. So let me say that it is our pleasure today to have a webinar moderator who's going to do a fantastic job walking us through the rheology essentials for pharma scientists. The second part of which is practicalities of measurements. It's my privilege to be able to introduce Dr. Nisarg Modi. He's the Director of R&D at Transdermal Research Farm Laboratories. He completed his Bachelor of Pharmacy from Sardar Patel University in India and received an MS and PhD from the Division of Pharmaceutical Sciences at the Brooklyn campus of Long Island University, or LIU. During his tenure at LIU, he worked on the development of semi-solid products and the evaluation of their internal structural properties using rheology. Dr. Modi has expertise in developing transdermal delivery systems, or TDS, for new chemical entities as well as for drug products approved through the 505(b)(2) pathway. In addition to his remarkable innovation and deep expertise in TDS and transdermal product development, he also has a fascinating breadth of experience in other areas of pharmaceutical sciences. In fact, in addition to the development of successful commercial products, his innovative work in transdermal drug delivery has earned several patents. Dr. Modi is also the secretary of the AAPS Topical and Transdermal community, and without the enormous amount of hard work and dedication to our community that he's given us, I can honestly say we would probably never have been able to produce an entire year of fantastic monthly webinars that we've consistently been able to bring to you. So we're very fortunate to have Dr. Modi with us to moderate today's webinar, and I hope you'll join me in welcoming him. Dr. Modi?
Thank you, Dr. Rane, for a nice introduction. Thank you very much. It is my pleasure to introduce Neil Cunningham, today's presenter. Neil is a very highly experienced consultant in the application of rheology and viscosity testing techniques across a range of industry, including pharmaceutical, medical device, and consumer healthcare. Since he started in this field in 1995, he has successfully carried a niche consultancy for the world's leading manufacturer and has trained and advised thousands of scientific personnel in the principal and practical application of rheology and viscosity testing. Over that time, he has gained an intimate knowledge of the most popular rheometer and viscometer in the marketplace, as well as a unique breadth of understanding of the need of industry. Throughout his career, Neil has also worked with the instrument manufacturer, including TA Instrument and Brookfield, providing consulting, instrument design, webinar, and training courses. Neil has a reputation for his accessible, enthusiastic approach to rheology and his ability to convey the seemingly impenetrable concept to even the most non-technical persons. Neil is the CEO of the Center for Industrial Rheology, which he founded in 2012 and which has grown to be one of the world's most trusted and respected laboratories, providing research services, contract, sample analysis, training, and consulting services on rheology. We are very fortunate to have Neil with us to help us explore Rheology Essentials for Pharmaceutical Scientists, a very special two-part webinar. We had a previous webinar yesterday on theory of the rheology, and today he is providing a webinar on a practical application of viscosity and rheological measurement. Please join me to welcoming Neil Cunningham. Neil?
Fantastic. Thank you very much, Dr. Modi, and thank you very much, Dr. Rani. Thank you, Dr. Modi, for that great introduction. I really appreciate it. Good morning, good afternoon, everybody. It's really nice to be here again. I hope you enjoyed yesterday's session and last month's session if you watched it first time round. So today I'm going to be talking about practicalities, really, and how do we actually go about measuring viscosity, measuring rheology, and then using that information for our formulation, quality control, and process development needs. I'm going to be packing a lot of content in over the next half an hour here. So please, if there's anything here that you don't catch first time round, grab my email address, neil@rheologylab.com. Don't hesitate to contact me afterwards. More than happy to talk you through this one-on-one, however you want, really. Okay, so let's get started then. Do you remember from yesterday, I introduced a concept of structured liquids. So the concept the concept that we have this model of materials that we can call structured liquids. These are suspensions and emulsions. They typically follow this sort of behavior where we could describe them as solids that could be made to flow. And here we can see the example of the mayonnaise I showed yesterday, showing very obvious solid-like behavior, that elastic bounce. But then when we apply sufficient stress to exceed the yield stress, then we can get that to flow. So we know then that we've got a number of properties that we need to capture and quantify to accurately describe these materials then. When the product is in its liquid-like behavior, viscosity is a relevant metric there, and viscosity across relevant conditions. But this solid-like behavior, then we have this viscoelastic mass that has a rigidity and a yield stress, strength and rigidity, and it also displays these thixotropic behaviors there, so the rate of breakdown and the rate of recovery after shear. So in order to accurately get a good representation of a material, it's highly necessary to be able to capture these metrics. We're going to start at the top with viscosity. Now, most people's experience of viscosity really starts with the simple Brookfield type viscometer, and these are workhorses of laboratories all over the world. Really, they're simple to use, they're low cost. And you basically dip the spindle in your sample, press a button, and get a number from there. But the apparent simplicity of these belies a lot of opportunities, really, for potential confusion. And a lot of that confusion and issues with non-alignment of test methods really revolves around these, what I call the Brookfield questions, or the Viscometer questions here, really. These are the typical issues that are likely to lead to two labs measuring the same product and getting different results here. So it's important that we can try and nail down a lot of these issues here, really. So let's just look at how the instrument operates. It's a pretty basic principle. You have a motor in the head of the instrument. That motor turns a shaft. The shaft is connected via effectively a calibrated spiral spring, which is a part of the torque transducer mechanism, to the spindle that then dips into the sample. So the sample offers a resistance to rotation, and the deflection of the spring is measured by the torque transducer there. And the torque that we get, this is the percentage value that you see displayed on the viscometer, is then converted into a viscosity reading here. So pretty straightforward approach. There are a range of different torque ranges that are available for the viscometer. Okay, and so at the business end here, where we're in contact with a sample, then there are a range of different spindles and accessories that can be used in certain situations. The standard set of spindles are these two. So at the top here, we have a set of RV spindles, and below that, a set of LV spindles. Okay, so these are typically provided with the kind of the mid-range viscosity model of the instrument and the low viscosity model here. But then we've got a few more specialist things. T-bar and helipath attachments here, I'm going to talk about in a moment. And then when we want to get into defined shear measurements, we have cone and plate and small sample adapter approaches here. So when we come to actually selecting and developing a test method, there are two situations. If we've got an established test method that we're working to, then we need to align on all of these aspects here, really. Okay. If we're developing a new method, then we've got more of a free rein there. And what we're typically doing there is we need to look at the temperature and the conditioning, the preconditioning of the sample before we take the measurement. And then we need to select a test method that's going to drive the viscometer more or less in its middle range, really. So there's a sweet spot of torque of around about 40% to 70% for samples that are in the middle of the spec. And this is a good range to go for here, really. And then we typically take a measurement after maybe 30 to 60 seconds. Now, I've given you a bit of a simplified approach here, really. And there's quite a lot of areas to get into. But a couple of things that we need to specifically watch out for here with this type of test is, firstly, what's the impact of having a thixotropic sample? So what happens if a sample's viscosity changes throughout the measurement? I've got an example here. We're taking a measurement. We've just got the motor going. It's running at 10 RPM here. And you can see we're measuring around about 350,000 centipoise viscosity here. But the value is now dropping. Okay, and this is actually a fairly mild drop. Occasionally you can take these measurements and you get a pretty significant drop in viscosity with time there. The other thing that happened here is that right at the beginning of the test, the value ramps up. This is where that calibrated clock spring is winding up to the full torque here. And then it's slowly decreasing as the sample undergoes a thixotropic breakdown. And so what's happening to our sample is what we can see in the plot here. It starts off with a high viscosity and then is dropping down towards what we call an equilibrium viscosity. An equilibrium viscosity, you could think of this as a kind of a baseline viscosity for that sample under those shear conditions there. This raises a question, when do you take a reading then? And it's important here to understand that if you take a reading early on in the measurement, you're going to get something that's more representative of the product in the condition it was in when you started to measure it. But that's going to be a much less repeatable measurement as well, really. And the longer you take to effectively pre-shear the sample, the more repeatable those measurements become. Okay, so thixotropy is one thing to take into account. Thixotropy can be thought of as a breakdown of structure. When you do have a highly structured sample, then if you don't have sufficient accessories, you get into using spindle number 7 on the RV set, or maybe spindle number 4 on the LV set of spindles, what we call the spindle of last resort. This is the only spindle you can poke into your sample without leaving a great big hole and a big air gap when you're taking a measurement. And that's not a satisfactory situation there. So it's pretty obvious here that we need to take another approach. We get many samples in my lab arriving with holes poked in the top of them like this. And then we know we're dealing with a structured sample and we need to take another approach. There's a simple attachment that can go onto a viscometer, the helipath and T-bar attachment. The T-bar is a type of spindle that is exactly that. It has a horizontal bar across it. And the helipath is this unit at the back here. What this does is this drives the instrument down slowly while the T-bar spirals its way through the sample. This is really useful because what this is doing then is it's allowing the viscometer to cut its way into fresh sample right the way throughout the measurement. And so what we're really doing here is we're kind of taking a measurement of a structural strength. There's some unknown kind of shear conditions going on here, but it's a simple method of measuring a kind of a gel strength, a structure strength, kind of related to a yield stress effectively, really, rather than a viscosity. But it's a useful method if you're working on a low budget and you've got a standard viscometer and you just want to try and extend the range of it. You have to be careful though. You have to take multiple measurements on a single down sweep and then take a mean of those values. I've got a fantastic bit of video that my chap Joey here captured a few weeks ago. This is a T-bar working its way down through this is actually a foam bath, and it's filled with little gold particles and air bubbles. And you could see the T-bar working its way down through and smashing into the bubbles. Now, whenever you hit one of those bubbles or a particle in there, that's going to give you a pretty significant jump in the results that you get. So that's why it's always necessary then to run this for around about maybe a minute and take measurements maybe 10 seconds apart and then get a mean value of those measurements. The bigger the T-bar you can use, if you can use a T-bar A or B rather than the E or the F, then that's also going to give you a much more repeatable and more repeatable result on this. Okay then. Okay. So give you a quick kind of introduction to some of the basic approaches that we use, simple viscometry. But then, if we really want to understand the viscosity behavior of our product over wider shear conditions, then we need to start to get into some more high-performance rheometry approaches here, really. And you can see some of the requirements of the FDA here on the right-hand side, and the EMA when we're looking at, for example, generic topicals. We're looking for a complete sweep that includes upper and lower plateaus here. So zero shear and high shear viscosity plateaus that I talked about yesterday here. In order to get these, you're going to need to be able to measure over a very wide range of shear rates and a very wide range of shear stresses. And then you need to get into high-performance rheometry. So up here we can see one of three high-performance rheometers that I've got in the lab here. And these instruments, the good thing about these is that you have a high-performance motor that's got an enormous torque range, and everything is mounted on a cushion of air. So the only contact between the whole rotating element here and the outside world is through the sample at the bottom of the plate here. Really. Okay, so I used to sell these instruments, actually. I used to be a sales guy and sell these things. And if I knew then what I know now about the performance of these instruments, I would have probably sold a whole load more rheometers. I just want to kind of illustrate really how different these are from the simple sort of viscometry approaches. Here's a pseudocreme sample, so a simple cream here. We've measured it across a range of shear rates. We've plotted this on a linear scaling. Now, if you're doing some more kind of basic viscometry, you would be looking at this, looking at these data points here, and you can see a few data points down to here, but they're sitting pretty much on the, right on the x-axis, and then all of a sudden there's this stuff going on up here at the low shear rates here, very low shear rates. But there's something interesting going on. We're not quite sure what's happening here. If we replot that on a logarithmic scaling, we get a completely different picture. And in fact, just to add a bit of perspective here, I'm going to replot it further. I'm going to put RPM. This is the RPM that the rheometer is working at when we took these measurements here. Okay, so what we can then see is that the data points here are these upper three data points here. Right down here, we've got a data point that's at 1/100th of an RPM. So this is the low limit for most viscometers on the marketplace here. But then all the action here is really occurring at below that limit here. But just to give you an indication of the kind of performance you get out of these rheometers, here we're taking a measurement that would take only 30 seconds to 60 seconds to take the actual measurement, the data point. If we let it rotate for a whole for one revolution, it's going to take a whole week to gather that data point. And if you get right down into the zero shear plateau here, then this final data point, if we let that run for a whole revolution, we're going to be hanging on for 3 months for it to do that. So I think this really kind of puts some perspective on what you get when you're paying $60,000, $70,000 for a rheometer system here. So, okay, so when we run viscosity profiles in my lab here, we're typically running one of these three tests here. And we're running either steady state sweeps, where each data point is gathered under what we call equilibrium conditions. We apply the stress, we allow the sample to reach an equilibrium viscosity, and then we plot the data. And then we go to the next value, the next value, the next value. We typically run that as an upsweep here. This is great for capturing zero shear and capturing the yielding behaviors. If we want a quick feel of a sample, then we can disregard the zero shear, at least initially, and we can just do a quick rapid shear rate sweep. And again, viscosity versus shear rate here. And then the other thing we could do is if we want to smoke out some thixotropic behavior, then we can ramp these shear rates rapidly up, maybe have a peak hold, and then come down again. And what we've done here is plot stress versus shear rate. And then we're looking for hysteresis and a hysteresis loop here to indicate the presence of thixotropy, this slowness in recovering after shear. Okay, so viscosity profiling often raises two questions once you get into high-performance rheometry. And the questions that I typically get asked there are, well, firstly, what's the difference between a controlled stress and a controlled rate measurement? Should I be running the viscometer, or the rheometer, in a controlled stress mode or a controlled rate mode? And one way to think about that is if you think about actual flow processes, you can divide up, you can split out actual flow processes into what I call free flows and forced flows. So a forced flow, imagine if you were painting a wall, then you're spreading the paint with the paintbrush, and that's a forced flow situation. You're in control, and you can determine how quickly you want that paint to be moving as it's spread across the wall. As soon as you stop painting, we return back to a free flow. Now gravity and maybe surface tension and capillary forces take over, and now the product, the paint itself, is, you could think about it as being in control, really. Gravity and these capillary forces are applying stresses, but the rheology of the paint determines what's going to go on from there. And so these are analogous to controlled stress tests, the free flow situation, and controlled rate tests, the force flow situation. So this is a useful approach, a useful way to think about this. You think about it when you're, imagine you're making, say, a bechamel sauce. Then there's two ways of assessing the viscosity of that. You can look at how well the product clings to the spoon. So here we're getting gravity to apply a stress to it, and we're measuring the shear rate. We're assessing the flow rate here. This is a controlled stress process. Alternatively, we can stir the sauce, and we're stirring at a certain rate, and we're measuring the resistance. So we're stirring at a certain shear rate, you can think of that, and the shear stress is what's being measured there, really. Okay then, those controlled stress methods, these delicate methods, they tend to be good for these structural assessments then, controlled rate, really better for understanding the big driven flows. It could be injecting, spreading, pumping, mixing, filling. Okay, so when we have ourselves a high performance rheometer, there's a number of ways in which the instrument can interact with the sample. And the most common methods we use here are the cone plate, the parallel plate, and the concentric cylinder approach. I'm not going to go into any great depth with these at this stage really, but cone plate is a nice simple system in that it generates a uniform rate of shear across the sample, across the entire sample. The downside of the cone plate is that you can't roughen the surface easily and maintain the geometry needed to maintain accuracy here. So then we often use the parallel plate. The parallel plate is great, and we probably use the parallel plate more than anything else in my lab here. With the parallel plate then, you can roughen these surfaces, so that could be sandblasting or hatching, and we'll come on to that a little bit later when I talk about slip. We can also adjust the gap here so that we can help to preserve the structure of the sample when we are loading the sample and bringing the plate down to the final gap ready for the measurement. Okay, there are other situations when maybe we want to use a concentric cylinder, or what we call a cup and bob. So here we have a rotating element inside a temperature controlled jacket. The sample sits inside of this, and the shear field is set up when this rotates between the two here. This is pretty useful because you have the air interface here. If you've got a sample that's likely to form a skin, then that skin, it will be less problematic than in a cone and plate or parallel plate situation where that will occur at the outside edge where the moment is at its greatest here. But the concentric cylinder system can be quite difficult. It's hard to clean. It's hard to fill, and you can completely trash your sample structure when you're loading it in there. Okay then. Okay, so we talked so far really about viscosity, and it started off with the viscometers and then getting into high performance viscometry and using that for extended shear sweeping. But what we also get with high performance rheometry is we get to look at some of the other aspects of a material's behavior. I love this saying by Abraham Maslow: if all you have is a hammer, everything tends to look like a nail. If all you have is an instrument for measuring liquids, you do have a habit of thinking about everything as being a liquid, really. And this is then really where the capabilities of primarily oscillation testing start to show their value here. Then. And I talked about oscillation testing yesterday. Remember that with oscillation testing, we're applying these very small perturbations to a sample structure, to a sample, and we're just looking at the elastic response or otherwise then. And from that response, from the resulting strain to our applied stresses, we can calculate out a modulus, the rigidity of the sample, and then we've also got this phase angle value here, this scale that's got complete elastic solids on at 0 degrees, complete viscous or inelastic materials at 90 degrees. And then we can use that in conjunction with the complex modulus to resolve out our storage modulus and loss moduli. And then I talked yesterday about oscillatory stress sweep tests and how useful they are. And these are really fantastic tests. They're great, they're repeatable, they're quick to run. There are a number of different ways of capturing the data from them. For example, we often want to quantify yielding behavior from these. And so we can look at, we're ramping up the stress here, and we can look at how our sample structure breaks down. And we can see the breakdown in structure by the rising phase angle. And then we can start to quantify. We could quantify what we call the linear viscoelastic limit, the first visible deviation from the plateau, looking here in the storage modulus here. Or we can look at the more significant yield events occurring. If we're interested in squeezing something from a tube or scooping something from a tub, then the linear viscoelastic region is not such a relevant metric to measure here. We really want to be looking at something like the flow point, the crossover point, where we're getting these great big changes in the material structure there. Okay then, and then working below the yield and maintaining our condition below the yield, then we can sweep the frequency and we can start to capture the viscoelastic fingerprint of our material. And there's a couple of different ways of presenting this data, but the most common approach is to present storage modulus and loss modulus as a function of frequency here. And what we can see here are two hyaluronic acid formulations here. The filled circles here, these are a dermal filler, actually, and what we can see is that we get a very elastic dominant behavior here, where the storage modulus is higher than the loss modulus throughout that range of frequencies. And then the other product with the open squares here, this is an ophthalmic viscosurgical gel, an OVD, that's used in cataract surgery. And these tend to be a lot more gloopier, slimier, stringy, this sort of thing. And that gives you this characteristic crossover behavior where we only see the elastic dominance at short time scales, high frequency oscillations. Then we get this crossover down into liquid dominant behavior here. We can see the same thing when we plot the phase angle as a function of frequency. We see very clearly that elastic dominance in the dermal filler compared to this relaxability that we're seeing in the OVD, the ophthalmic gel here. Okay then. Now another great insight that oscillation testing can bring you is in understanding thixotropy. And so it gives you a kind of a new insight into thixotropic behavior. So rather than doing the up-down shear sweeps, or even the stepped shear sweeps, we can actually work at very small strain oscillations. And we could start off with a conditioning period, and you can see for both of these samples here, we can see step 1 is a low strain conditioning. Phase angle is low. Remember, low phase angles indicate intact structure. We then hit the sample with a large strain deformation, and that disrupts the structure. And then we can see for step 2, the phase angle is much higher there, so that indicates less elasticity. And then what we can see for the slow recovering, for the more thixotropic material, we can see that long, slow recovery as phase angle makes its way back down towards the initial values here, really. So this is it gives you another angle, another useful insight on thixotropic behavior. Okay, then. One more thing we need to look at is we need to look at what can cause problems for you when you're taking measurements. Many viscometers and rheometers are provided with some very nice, smooth, shiny surfaces, and that's often the last thing that you want, really, when you're performing rheology testing. In rheology, we want to encourage bulk shearing, and if we have smooth surfaces, that can work against that. Here we can see on this smooth glass plate, we can see this cream just sliding down the plate, and that's the last thing we want to be occurring up against our spindles and our measuring systems on the rheometer. So we can, and that can really cause you some problems in your data here. So we can deal with this in various ways. We can use roughened surfaces and crosshatched surfaces and sandblasted surfaces to grip the sample and to eliminate that. And you can see some very obvious differences here. Okay, then. Okay. Finally, I just want to talk about how do we, we've gathered a whole bunch of really, really nice data. We've avoided the artifacts or dealt with the artifacts. How do we present that in a way that enables us to exploit that data and get some real value and be able to communicate that data nice and easy to other stakeholders? Here's a typical example of a rather simplified approach that we take, excuse me, for many topical products. Here we're looking at the consumer experience of a product. We're divided up into 3 stages. We've got an initial stage where we take the product out of a tub or we squeeze it out of a tube. The middle stage where we're applying it, and then the late stage where we're really rubbing it in, and we're looking at an element of afterfeel here. And we can measure yield stress, viscosity at application shear rates, and then coefficient of friction. So we're moving outside of rheology a little bit here into tribology here, but this is really useful. And then what we've done here is we've got a whole range of different topical products here, and we've measured the yield stress, the viscosity at a defined shear rate, and the coefficient of friction, and we can plot those together, and we can start to use this to generate this rheological space that gives us an indication of similarities or dissimilarities between our products. And then finally, if these 3 dimensions aren't enough, we could start to get into some really nice statistical techniques then for understanding data better and assessing similarity to certain benchmarks and reference products. I showed this data yesterday. These are some oscillatory temperature sweeps showing highly nuanced results here. And so what we've done here is we've used 2 approaches. We've used a machine learning clustering algorithm here, and we've used principal components analysis to decode and deconstruct this data and to group this and cluster this. And we've taken the clusters that are delivered by the machine learning algorithm, and we've colored those data points in according to those clusters in the PCA plot, the principal components analysis plot. And we can see a really nice agreement, a pretty good agreement between those. So we use these as a cross-check here, and we've colored the plots accordingly to the same coloring. And you can see that we're getting a pretty decent recognition of the patterns and the similarities here. This is fantastic if you're looking at something like a white soft paraffin. You're looking at a second source of white soft paraffin or some complex mixture of of products, waxes and oils and those sort of things. Okay then. Okay, so just to wrap this up then, what we can see then is we could take this structured liquid model, very useful. We measure the metrics that describe the different aspects of that structured liquid behavior for the product, the soft solid behavior and the liquid-like behavior. There are many viscometers out in the market that can give us a simple comparative measurement. But if we really want to get into get a really good solid understanding of our product and maybe references and new versus old and reformulations, then we really need to get serious about rheological profiling and start to look at structure and viscoelasticity and broad shear rate and broad shear stress viscosity profiling. Okay, so I hope that's been useful. As I say, it's a bit of a whistle-stop tour there, really. Please don't hesitate to get in touch with me if you want me to go through anything again. And I'm looking forward to answering a few nice questions now as well. Thank you very much. Over to you, Dr. Muddy.